Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article, Nivolumab in Patients with Relapsed or Refractory Hematologic Malignancy, Preliminary Results of a Phase 1b Study by Lesokhin et al. My name is Jason Weston, and I am an assistant professor at the MD Anderson Cancer Center in Houston, Texas. My oncologic specialty is the treatment of patients with lymphomas. One of nature's greatest killing machines is an activated T cell. Once the T cell receptor interacts with antigenic peptides presented in the major histocompatibility complex, cells expressing the target protein should beware. Unless an inhibitory co-receptor, like programmed Death1, PD1, or cytotoxic T-cell lymphocyte-associated protein 4, CTLA-4, are involved. These proteins, often referred to as immune checkpoints, are powerful regulators against runaway T-cell function and play a critical role in preventing autoimmunity. Lymphoma cells have a complex interaction with their benign immune counterparts, which includes co-opting the interaction of PD1 and its ligands to avoid immune equilibrium or elimination. Therapies directed at immune checkpoints have shown remarkable progress across a wide number of cancer types, including melanoma, lung cancer, renal cancer, and now hematologic malignancies. Potent signs of the power of immune-directed therapies for lymphomas came from trials of ipilimumab, lenalidomide, and allogeneic stem cell transplantation. Ipilimumab, a CTLA-4 inhibitor, demonstrated responses in relapsed B-cell non-Hodgkin lymphoma. Lenalidomide, an immunomodulatory agent, has shown impressive activity across a wide spectrum of hematologic malignancies, including myeloma and both indolent and aggressive lymphomas, both by direct anti-tumor effect and by immune modulation. Lastly, the graft versus lymphoma effect of an allogeneic stem cell transplant clearly establishes the power of the immune system to battle lymphoid malignancies. These data, in addition to the heavy PD-1 positive T-cell infiltration observed in many lymphomas, led to trials of nivolumab and pembrolizumab, which are fully human immunoglobulin G4 monoclonal antibodies that target membrane-bound PD-1. The highly anticipated initial results of the Phase 1 trials of these agents were presented at the 2014 ASH annual meeting, and they surpassed expectations, especially for Hodgkin lymphoma, with an 87% overall response rate for nivolumab. Based upon these early data, along with ongoing confirmatory trials, the FDA granted approval of nivolumab for patients with Hodgkin lymphoma relapsed after autologous stem cell transplant and brentuximab. The publication, which is the focus of this podcast, led by Dr. Lesokhin and colleagues, presents the data for nivolumab with other hematologic malignancies. The aim of this Phase 1 open-label dose escalation cohort expansion study was to determine the safety of nivolumab in hematologic malignancies, with secondary objectives including correlation of abnormalities of the PD-L1 and L2 loci and protein expression to evaluate for correlation with response. Patients with relapsed or refractory lymphomas and multiple myeloma were eligible if they were at least 18 years of age, had an ECOG performance status of 0 or 1, measurable or detectable disease, and had at least one prior therapy. Patients were excluded if they had CNS involvement, other malignancies or autoimmune disease, prior allogeneic or solid organ transplant, or prior checkpoint inhibitor therapy. As the results for Hodgkin lymphoma have been previously reported by Dr. Ansel et al. In 2015, this article is focused on the results for non-Hodgkin lymphoma and multiple myeloma. Patients were treated with nivolumab at 1 or 3 milligrams per kilogram every 2 weeks for up to 2 years, stopping early for progression, toxicity, or a complete response. If a patient stopped early with at least stable disease, they could be retreated if progression occurred within 1 year. No maximal dose was found, and thus all patients treated in expansion received 3 milligrams per kilogram. A total of 81 patients were treated, including 27 with myeloma, 31 with B-cell non-Hodgkin lymphoma, and 23 with T-cell non-Hodgkin lymphoma. Of the B-cell non-Hodgkin lymphoma, 11 had diffuse large B-cell lymphoma, and 10 had follicular lymphoma. Patients had a median of 3 prior therapies, with greater than 70% of myeloma, T-cell, diffuse large B-cell lymphoma, and follicular lymphoma patients having 2 or more prior therapies. 96% of patients treated experienced at least one adverse event, and 65% had an event deemed drug-related, including 18% grade 3, 2% grade 4, and 1% grade 5. Immune-mediated events occurred in 34%, with 4% grade 3 or higher pneumonitis. 46% of immune adverse events resolved without treatment or treatment interruption. Of the patients who required therapy for immune events, 66% were able to continue therapy with nivolumab. Only 15% of treated patients discontinued nivolumab due to drug-related adverse events. 1 grade 5 toxicity occurred on study, a case of pneumonitis occurring after a single dose in a heavily pretreated patient with a history of asthma and chronic cough. Responses in the myeloma patients were modest, with 63% achieving treatment benefit with stabilization of disease lasting a median of 11 weeks. A patient who required holding nivolumab for radiotherapy of a symptomatic plasmacytoma entered a durable remission, potentially representing a manifestation of the abscopal effect. In the B-cell lymphoma subsets of follicular and diffuse large B-cell lymphoma, the overall response rates were 40% and 36% respectively. The overall response rate for T-cell lymphoma patients was 17%, with responses lasting greater than 24 weeks in 3 of the 4 responders. Of the 27 patients with tissue available for interrogation, only 3 were found to have a 9p24 alteration, in stark contrast with previous findings in patients with Hodgkin lymphoma. A patient with mycosis fungoides had a FISH-detected anomaly which resulted in overexpression of PDL2 on malignant cells and experienced a durable partial response lasting 47 weeks. The other patients with 9p24 alterations had low-level expression of PDL1 or L2 and achieved a partial response and stable disease. In the remaining cases, immunohistochemistry failed to detect a significant expression of PDL1 or L2 on malignant or benign microenvironmental cells. The authors should be congratulated for an important clinical trial, establishing that the PD-1 antibody nivolumab is generally well tolerated, has similar adverse events to those seen in prior trials of nivolumab with solid tumors and Hodgkin lymphoma, and results in clinical activity in a variety of subtypes of hematologic malignancies. Despite significant effort by the investigators, no correlation between response was identified with aberrant expression of PD-1 or its ligands, which could be influenced by the relatively small sample size. The search for a biomarker, critically important for helping identify patients most likely to receive benefit with checkpoint inhibitor therapy, will continue in upcoming Phase 2 trials in follicular and diffuse large B-cell lymphoma. The desire to harness the power of the immune system against cancer has been a long-standing dream, dating back at least to Dr. Coley and his injected toxic brew. Recently, a powerful group of new anti-cancer weapons have been developed, which now includes nivolumab. The safety data reported here is encouraging, but immune adverse events are not insignificant, especially in light of the capacity of other targeted agents to impact immune function. The efficacy reported in the manuscript is not overwhelming, which is not terribly surprising for a single-agent therapy for heavily pretreated hematologic malignancies, but is in contrast with prior remarkable data for Hodgkin lymphoma. However, this trial establishes a baseline for the safety and efficacy data for nivolumab monotherapy and should result in numerous studies combining checkpoint inhibitors with other targeted therapies, either directed against hematologic malignancy cells or further augmenting the immune response. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.